Hi, we were talking about the replication and how it is maintained in eukaryotes and prokaryotes. We only mentioned the proteins and the enzymes that take place in the replication process. Now we will look into the other properties of the polymerase itself. So it doesn't only synthesize, but also it does some editing, which is called the proofreading. When scientists looked into the E. coli during the replication process, they saw a shape like this, and it is called a lariat shape. Let me draw it like this. And first they thought that the replication continues in the both sides, which is correct, but also in the same direction for both strands, which is incorrect now that we know of. In an opening like this, which is called an origin, DNA synthesis starts, and if on this strand it goes like this, then on the strand it will go like this in pieces, and also in this direction it continues in order to make a copy of this outer ring and the inner ring. So now we call these fragments as Okazaki fragments, and this strand is the lagging strand. Also in linear chromosomes, if we have a chromosome like this, if this synthesis is continuous on this side, then on this side it will be continued like this, and here it will be the lagging strand. So the directions would be 5' to 3' for this strand, and 5' to 3' for these Okazaki fragments, 5 to 3 for this one, 3 to 5 for this one. When the polymerase continues to make the synthesis for both strands, like this, it adds the nucleotides one by one, and it will be complementary to the other strand. The polymerase in a very basic structure has these pockets inside it, and this is the polymerase itself, and the DNA strands will be coming like in this direction. This is the template. And this one is the newly synthesized strand. The processing will be performed in this pocket, which is the processing pocket or polymerizing pocket. Yes, this is the polymerizing pocket. It always adds the correct nucleotide, which will base pair with the template strand. So if there is an adenine in the template strand, the polymerase will add thymine and nothing else. However, not every nothing in the biological world is perfect. The polymerase also makes mistakes. The mistake rate is very, very low. It is like 1 in 10 to 10. Like this is a huge number, 10 to 10. But because the replication process is a continuous process and it takes place all over the lifetime of a cell, then this means something, and all over the lifetime of a cell, the cell may accumulate a couple of mutations. And also, this is after the proofreading. Also, the polymerase itself has a proofreading mechanism. Suppose that there is a T here and it added A, and there is a G here, and instead of C, a nucleotide came, adenine nucleotide came. What happens in this situation is, because the polymerase has a groove and it detects the shape of the nucleotides, and two purines, because the guanine and adenine are both double ring structures, it will occupy a larger space, and then the polymerase detects this and directs the synthesis into the editing pocket. In the editing pocket, the incorrect nucleotide is removed and the correct nucleotide is replaced by it. So this is part of the process called proofreading process. Proofreading is different from the repair mechanisms. Repair mechanisms, which we will see in another chapter, but shortly today. Repairs are scans the genome. Like in the cell cycle, there are the interface is divided into three parts: G1, S, and G2. And before the synthesis of the DNA, the repair systems scan the whole genome to find mistakes in the genome. And if they find a mistake, then they change that nucleotide, and it is called a repair system. But the proofreading system is that directly maintained by the polymerase itself. It checks itself. It is the same process, like you write a paper and simultaneously you check for the mistakes, or you reread again and check for the mistakes. Also, it can scan the genome while the replication continues. So we can call that the proofreading mechanisms are during the replication and during S phase only in the cell. But the repair mechanism can happen in any time in the cell cycle. Proofreading mechanisms activities can be divided into exonucleases. Now we have a word exonuclease. This means if you have a strand, DNA strand, and this part is newly synthesized, so it is an open end, and it is an open 3' end. If it makes a correction in this part, in this area, then it calls the 3' to 5' exonuclease. Because the nucleotides are added in here, and if there is a incorrectly added nucleotide, then, and this has a 3' OH group, it is removed from here. The T is removed. Now we have another 3' end, which is now not occupied, not synthesized. So this is called the 3' to 5'. If, on the other hand, suppose that there is a primer sequence here. In the beginning of the replication, we have the primer sequences, and they have to be removed after the replication process, because the primers are made up of RNA. Then a polymerase comes and chews from here, like a Pac-Man, it chews from here. This is called the 5' to 3' exonuclease. These are embedded into the polymerases. Like a polymerase can have a 3' to 5' polymerase exonuclease activity. They always have the 5' to 3' polymerase activity. All of the polymerases, but some polymerases have this and some polymerases have this activity. There is another type of nuclease which is called endonuclease. The polymerases do not have any endonuclease activity. Endonucleases are separate enzymes. They cut through the strand, in the middle of a strand. They do not require an opponent, like they don't require a 5' opponent or a 3' opponent. They cut from in the middle of the strand, and they are called endonucleases. Now this is if it is a part of the polymerase enzyme, then it will have an exonuclease activity, and it is called a proofreading. So the mistakes 1 in 10 to 10 comes after the proofreading activity. For the remaining mistakes, we have DNA repair systems. Before looking at the DNA repair systems or how mistakes are corrected during the synthesis itself, I want to draw a whole picture of the polymerase synthesis polymerase system. Before that, I want to clarify the cell cycle. Now the cell cycle is something like this. Mitosis takes place in here, and all the remaining is interphase. In this mitosis we have prophase, metaphase, anaphase and telophase. The interphase is divided into G1, S and G2. In the S phase we have the DNA replication. And all the proofreading activity and the repair system that I will talk about in a minute are taking place in this S phase. The other DNA repair systems work in the other parts of the cell cycle, either G2 or G1. During the M phase, there is no activity because the nucleus is nucleus is dispersed, and the chromosomes are packed into compact shapes. So anything that has to do with the chromosomes itself cannot be done during mitosis. So they can be done in G1 or G2, and there are checkpoints. Here there is a checkpoint, and here there is another checkpoint. In these checkpoints, DNA repair systems scan the whole chromosomes and look if there is a mutation or not. If there are mutations, then they stop at that point, give a pause, and repair the damage. And before the DNA repair system approves the DNA, approves the chromosomes, the cell cannot go into the S phase, and cell cannot start the DNA replication process. And after the DNA replication process, there is another checkpoint which is maintained by the DNA repair systems, so that the chromosomes are intact and they are healthy and they are free of mutations, so that they can go into mitosis and divide into two daughter cells. The process takes place during S phase, are what I'm talking today, the proofreading mechanism and also a mismatch repair system. They work simultaneously with the DNA replication, and there's a good reason for that. First is the exonuclease activities are part of the polymerases, and polymerases are only active during the S phase. And the second, mismatch repair system requires an ongoing DNA synthesis. So actually I can talk about the mismatch repair first and then go into the whole scheme of the DNA replication. That will be better. The mismatch repair, or in full name, strand strand-directed mismatch repair. This takes place during the replication because this system has to discriminate between the newly synthesized strand and the old strand, because if there is a mistake, it has to be on the newly synthesized strand, not in the old strand, and the cell has to assume that the old strand is the correct one. And to do that, in eukaryotes, if there is a lagging strand, the nicks, if on the lagging strand there are nicks, so the polymerase is in here doing its job, and the lagging strand in here, leading strand is in here. In the lagging strand, the Okazaki fragments are not joined yet, so that is why the synthesis has to be ongoing, because these nicks will be repaired in a very short time. But before they are repaired, the mismatch repair system says that this is the newly synthesized strand, and if there is a mistake in here, this one has to be the wrong one and this one has to be the correct one. But on the leading strand, we do not know how it discriminates, and there are research going on, but it's funny that we still don't know why, how it discriminates between the newly strand and the old strand. In the bacteria, there are the bacterial chromosome. This is the chromosome, double helix. There are some methyl groups, and these methyl groups are on the sequences called GATC. When there is a guanine, adenine, thymine, and cytosine sequence like this, or in a pack like this, then an enzyme, methylase enzyme, recognizes this sequence and then adds some methyl group on top of the adenines. This is performed after the replication. The old strands have the methylations on itself, and this strand has the methylations as well. Now there will be the new synthesis. Because this strand is newly synthesized and because the synthesis replication is still ongoing, the newly strands are not methylated. So an enzyme, strand-directed mismatch repair enzyme, comes on top of the strands and see which one is methylated. So it recognizes the methylated strand and it assumes that the nucleotides on that strand are correct, and if there is a mismatch, it replaces the other strand. This is methylation based, and it works quite well for bacteria. Bacteria divide relatively very in a short time. The E. coli divides in every 30 minutes, so its replication is also very quick. But these mechanisms are in place to ensure that the replication is correct. Even bacteria has to make a correct replication, more or less. They have more mutations or more changes in their DNA than the eukaryotes, but still they have to maintain their genome. In the E. coli, researchers were randomly mutating the genes, and then they were looking at the effects. Like, for example, in each E. coli there's one mutation, and they were observing the behavior of this E. coli, how it survives, what does it do, what is missing, so on. They find out that when there is a group of genes, when mutated, the E. coli has lots of other mutations. So they say that we mutated only one gene. Why do we have like 10 other genes mutated in this E. coli? And they called these genes as mut genes, short for mutator. The mutator genes, like, it sounds like a bad thing, like the gene itself is causing the mutations or so on, but it's not. The simplest example would be the 3' to 5' exonuclease activity in the polymerase. If there is a mutation which blocks the activity of the exonuclease part of the enzyme, then the polymerase cannot correct its mistakes, and then you will see an accumulation of other mutations. So this is called a mutator. By definition, if you mutate one of the mut genes or mutator genes, then there will be lots of other mutations. So these genes are actually part of the repair system, and some of them are in the other repair systems, but some of them are working through the replication process, the strand-directed mismatch repair. Now I will show you a mechanism of it. In eukaryotes, I will check. It happens in both bacteria and eukaryotes, but in this example, it searches for nicks, so that it must be eukaryotic systems. So it works like this. Now there is the strand here. This is the mismatch. Suppose that it is adenine and guanine and occupying a larger space. And this is the newly synthesized strand, and this is the old strand. The system recognizes the newly synthesized strand from the nicks, and then which one is searching? The searching enzyme or protein is called the mutS. This one finds the mistake in the DNA, and scanning is done by another protein called mutL, or I'm not sure. Actually, in the book, it showed only these two, mutL and mutS, but as I remember, if it is named to the S and L, there must be other proteins from the alphabet, like A, B, C, D type of. So I know that there are lots of other proteins, but for simplicity, I think it showed only two proteins. These two proteins make this shape. I will draw in a minute. Now the mistake is in here, and mutS protein is in here, and mutL protein joins this shape, enables this shape, and then they cut the newly synthesized strand from both ends. Like, to include this one, they cut from, for example, from here and to here. They erase all that area so that there will be a missing strand. like this. So it doesn't just remove this part. It doesn't just take this incorrect base out. They cut a larger piece, including the mistake, including the mutation. So this is how they work, and then polymerase comes into this position because the replication is continuing and fills in this gap again so that the mistake is repaired. This is one way of working for the repair systems. In humans, actually, there are lots of mutator genes, and the mismatch, strand-directed mismatch repair has lots of proteins in its family. And there is a specific disease which includes these mutations. It is called HNPCC, hereditary non-polyposis colon cancer. This is a cancer, it's a colon cancer, and it is hereditary. So people acquire this mutation from their parents. Both parents can be carriers, and then the child can be homozygote for this mutation. And what it does, these people have mutations in the proteins of the mismatch repair system, and as a result, when they grow up, they cannot correct their mistakes. And this is true for all the cells in the body, but the mostly affected part is colon, because colon has an enormous rate of mitosis, especially in the children. Like there are trillions of cells are discarded every day, and new ones are made, so that there's a loss of replication going on. In other parts of our body, the mitosis rate is not that fast. Some organs, some tissues are very slow, some other tissues are faster. So because the replication is high in the colon, these cells in the colon accumulate mutations more. As a result, in a very early age, these people have the colon cancer. And as far as I know, it's the treatment options are limited because the mutations are in the repair system and they're coming from the birth. So the treatments are limited. And only one thing can be elimination of the other mutators, like external mutators like smoking or the sunlight. But because this is coming from the mistakes of the polymerase, the external factors do not have a role in this disease as well. Now we can draw the replication system. When we draw the strands, I was drawing like two strands going on each direction, but actually the replication process is a compact process. It takes place in a very compact structure. So there is a strand coming from here. This is the how it looks. This is the replication fork. This is the old duplex, and these are the newly synthesized duplexes. One is in the leading strand, so it's in pieces, and the other one is leading strand, so it is in one full strand. And the enzymes which look like will be here. We have the helicase. Helicase is the front runner in this process because it has to open up the helixes, and the helicase is large structure made up of like 6 subunits, so that if this is the strand, double strand, it holds the double strand like this and opens up. The catalytic site is in the middle of the cylindrical shape. And we will have the polymerases. The polymerase will be 2 polymerases in one fork. And here also polymerase. But in the other parts of the chromosome, there are other forks. Like this is one fork and there is another fork up in the other parts of the chromosome. Polymerase is in here, and there is a loader here. Clamp and clamp loader, and this is the I don't know how to say it, but it exactly looks like screw-like structure, and there's a metal, round metal with a hole in it, so that it can secure the place of the screwdriver. But I can show you with the Play-Dohs. This is our strand synthesis, and this is polymerase like this, and the clamp is just after the polymerase, so that it will secure its place. Because if the polymerase falls off from the strand, then the replication would be a slower process, but if you secure the place of the polymerase, then it can continue faster. And for this clamp, this is the clamp, there is a loader. A loader loads the clamp in here, and the loader is in here. It's a accessory protein type of protein. It loads the clamp, and this is, although this looks like an unnecessary protein, because it doesn't have any enzymatic activity, it just uploads the clamp, but it has a crucial role, because in the lagging strand, the polymerase synthesizes, and then it has to jump over the other part and then synthesize again. So because it's a lagging strand, it goes and synthesizes again, and then it goes and synthesizes again. So each time the clamp loader attaches the clamp and the polymerase to its new place. So that's the function of it. And also, if we have a single strand here, because this part is not synthesized yet, for example, or in here, actually it will take place in here. This is released, but the polymerase is not hooked onto this one and synthesized the strand, because this is a single strand and the cell doesn't like single strands. Single strand binding protein is SSBP attached to the single strand binding proteins. Now this clamp is, for example, there is a pCNA called proliferating cell nuclear antigen. This is a clamp for polymerase, and we usually use it as a marker for the cell replication. For example, if a cell is actively dividing or like replicating its DNA, it has to have pCNA. But if the cell is a very quiescent cell and it doesn't divide often, then pCNA expression would be very, very low. So we use this as a marker of the replication process. I think this is all for now. In the next chapter, I will continue into the replication, but this time how it is started and how it is ended. Because this is the continuing replication. We already looked at the continuing one. The start and the finish is highly regulated, and we will see how it is maintained.